Why, hello, friends. Good evening and welcome to this month's installment of Bris Science, presented by the University of Queensland. I'd just like to acknowledge the traditional owners of the land upon which we are gathered today and pay my respects to elders past, present, and emerging. And I acknowledge those amongst you in the hall today who are Indigenous. And if I'm an unfamiliar face, your usual MC, Joel, is unfortunately unavailable today, but I am a much younger, more good-looking version. I'd just like to direct you now to the screen. You will see a QR code being displayed, and here's how you can keep in touch with us throughout today's Bris Science talk. When you scan it, there's an option for you to interact and ask questions in that platform. And at the end of our speaker's talk, we're going to go through as many of those questions as we can. But if we cannot get to all of your questions, please stay behind and have a chat with our speaker. So, it gives me great pleasure to introduce you to tonight's topic. Tonight we ask an important question. We know that livestock, particularly ruminants, generate quite a significant amount of greenhouse gases. We also know that there's global pressure to reduce those greenhouse gases. So how can we achieve both, right? How can we reduce methane outputs from ruminants while still producing enough meat to satisfy consumer demands? What works? What doesn't work? Where's the body of work that we still need to uncover? Tonight's Bris Science lecture is presented to you by Dr. Sarah Meale. Sarah is Senior Lecturer in Animal Science at the University of Queensland and specializes in ruminant nutrition, gut microbiology, and sustainable production. Her research focuses on early life rumen programming and feed efficiency to reduce methane emissions, with work spanning lab studies to large-scale grazing and feedlot trials in partnership with industry. We're so privileged to be hearing from Sarah tonight, and if you would join me and make very welcome Sarah Meale.
Thank you. Well, hello, and thank you guys for coming out on a Monday night. It's very nice to be here. Most of you have probably heard methane, right? And probably not in a good way. So I'm hoping tonight that I can shed a little bit of light on what it is actually, why it might be a problem, and some ways we might be able to fix it, right? So stay tuned to the end, because it's not all doom and gloom. So, the global picture, right? 99% of global greenhouse gas emissions are carbon dioxide. 1% is methane. 1%. Of that, within Australia, it's broken down to 51% from agriculture, 25% from mining, and then an equal share from forestry and also from waste. Now, I hear some of you saying, okay, there's 1%. Why should I care? Unfortunately, the reality of methane is that it can warm the atmosphere 28 times greater than carbon dioxide. So that 1% is actually quite significant. It has a really big influence, but the world is not doomed. If you combine that with the fact that methane only stays in the atmosphere for 12 years, whereas carbon dioxide is over centuries, what that means is if we today stopped all methane emissions, in the next decade, we would see an effect. We can't do that with CO2. And in fact, reducing methane emissions is the single fastest way to reduce global warming. So why don't we act now? How do we actually do this? Agricultural emissions come almost entirely from ruminants, cows, sheep, and goats. The bigger the animal, the more methane they emit. So cows get a really, really bad rap. And the reason for this is they have a very unique digestive system, right? They can consume things that we can't. Grass, straw, hay, residues of crops that you and I cannot physically digest. Now, a ruminant can do this because it has a very specific digestive system. And in one of its chambers of its stomach, called the rumen, it acts as a fermentation vat. There are millions and millions of microbes that take that straw, grass, etc., and break it down into small molecules. The things up here that I've listed as acetate, propionate, and butyrate. That's what the animal can actually use to grow, right? Those are the good things. Now, ancillary to this, when they produce all of these wonderful things they can use, they also unfortunately produce byproducts, hydrogen and carbon dioxide. Now, if hydrogen accumulates in the rumen, it drops the pH. It makes it acidic. Microbes don't like that. In fact, they die. And if the microbes die, the animals die. Now, ruminants have evolved to avoid this really dramatic cascade, essentially, and they do this with the help of what's called methanogens, a very specific group of microbes that combine that hydrogen with CO2 and produce methane. And then it's burped out. The number of times I get told, but what about the farts, right? It's all about the burps. Today, we're trying to stop the burps. How on earth do we do that when those burps are crucial to the life of that animal? We are actually fighting evolution when we are trying to reduce methane, right? So keep that in mind. How do we get the best of both worlds? My wonderful MC said we need to feed the world. We need cows to do that. They eat, as I've said, that stuff that we can't eat. They graze lands that we can't grow crops on. We need them to produce the high protein milk and meat. Without them, the world cannot feed itself. It is simple as that. We have to have ruminants. We need to provide more food. We need to make them grow. But we have this bad thing called methane. How on earth might we reduce that? Right, so that's where my lab comes in. And it's something that we focus on, something that my group of students here today work really, really hard on. And we do that in a few different ways. We start out in the lab, and what we do here is we actually mimic what's going on in the rumen. So each one of those little bottles you can see has rumen fluid collected from a cow, mixed with some artificial saliva, and then in there, there's a little treatment, whether that be a different diet composition, so grain, hay, feedlot, whatever it might be, or an anti-methanogenic compound we're testing. We can rapidly screen hundreds of different treatments at a time. Now, primarily we're looking for methane. What does it do? Is it good or bad? But in this system, we can also see, does it have any negative effects? We don't want to just feed a product to an animal that might kill it. That's pretty bad, right? We want to make sure that we're reducing methane, but we might also have a positive effect on that animal as well, or at least no negative effect. So to give you an example of how we do this in practice, a recent study conducted by one of my PhD students looked at how many different products are either commercially available or really, really close to commercially available, and conducted a really extensive literature review. So trying to understand how they work, how might we include them in different diets. And we focus primarily on grass-based. Now, the reason I point this out is because the majority of our methane emissions come from our breeder herd. And what I mean by that is cows and their baby calves, right? They're eating a really highly fibrous diet, and they're there for a long time. So just with those two factors, they're producing more and more methane. We need to target that population to have the biggest effect. So once we did a literature review, we identified 28 different products. Now, we narrowed that down to 9, purely for feasibility and availability of products. Not everyone wants us to test everything all the time. And we tested ones with distinct modes of action. So I've listed them all up here for you on the screen. I'm not going to read through them. But you'll notice there's different doses, right? We didn't test everything at a standard dose of 1% adding it in. And the reason for that is their mode of action. They all work differently to reduce methane. And if we include them all at a standard dose, maybe they just don't work because there's not enough in there. So we used the manufacturer's recommended doses to find out what would the best one be. For our specific use, for grazing animals, what would the best product look like? And we narrowed it down to 2. So the 2 best ones we saw were a commercial essential oil blend, which gave us a really exciting 40% reduction in methane. That's a pretty big number, right? We also had a novel biochar. So biochar is what's formed when you take a piece of wood and you pyrolyze it or burn it at a really, really high temperature, and it produces something that has really large surface area and large pores. The theory being that those pores can then capture the gas rather than letting it be emitted by the animal. So we compared these 2 different products, and you can see pretty good results at the highest inclusion level, right? We had to put a lot in there to get an effect. Now, methane is one thing, but as I said before, we're also really interested in what does this do to the animal? And the primary measurement of that is digestibility. How much can the animal actually use of that feed? Now, unfortunately, when we looked at that really positive essential oil blend, we also saw quite a negative decrease in that digestibility. Now, what that means is the animal couldn't actually use the food. So theoretically, it's not going to grow as well, right? If we anticipate this is what's going to happen in the animal. And what's also really important to note is methane is directly linked to the breakdown, that digestibility of the diet. So by reducing digestibility of the diet, you inherently reduce methane, right? So not a good thing. We didn't see the same thing with the novel biochar. We saw a stable effect on digestibility, which is quite positive, right? No effect is also good. It means we didn't change how that product worked. We didn't change how the grass was digested. So this biochar was actually selected for further testing. We then looked at what happens if we put it in animals, right? So this is our next step. We start with a lab scale, and then we move on to what happens in an animal. And in this type of environment, we want to measure everything we possibly can, but we also want to control everything we possibly can. So you'll notice they're all black, they're all Angus, they're all the same breed, same weight, same sex, and they're all housed in almost exactly the same way. And in this case, they're individually housed, so we can also measure how much each animal ate, which is really important because methane, as I said, is directly linked to how much food they consume. If one animal eats a little bit less, well, they're going to produce less methane. It's just a fact. So we want to control for that factor as well. Now, in this study, we do what's called a metabolism trial. So as the name suggests, we're trying to understand exactly what is happening inside that animal when we feed this compound. So we had 3 different treatments. A control, you put nothing in, just feed it a regular diet. We had biochar, which is that charcoal-based product. And then we also had what we called an enriched biochar. Now that's basically the biochar where we've added stuff to it. So in this case, we added nitrate, which also has an ability to reduce methane, right? So we wanted to confirm what is the best mode of action, and can we actually enhance that beyond what we saw in our little lab-based study. So as I said, we measure intake. We want to know body weight. Are the animals growing? Are we meaning for them to grow? In some studies, we just want them to stay the same weight. We look at their rumen microbiome, and then those VFAs. Those are the molecules broken down the animal can actually use for growth. We want to see how much of those are available to the animal. And then, of course, methane, right? And in this case, we use what we call an SF6 tracer technique. And that's what I've shown in the picture here. So essentially what happens in this is the animal is given like a pill that has a slowly releasing gas. We can use that because it's released at a known rate. On the back of the animal, in the saddle component, is a cylinder that is evacuated of air, so it's slowly sucking in air. Now, if you trace that little tube, the orange thing, all the way down, it actually sits just above its nostrils. So as that animal goes about its day for a 24-hour period, we are capturing a component of the breath that comes out of that animal. We can then analyze that gas and understand how much methane was emitted over a 24-hour period without touching the animal. Pretty cool. So what happened? Intake. How much did the animals eat? Didn't change, which is a good thing, right? We don't want to reduce intake arbitrarily. But unfortunately, we didn't see that same 23% reduction of methane that we saw in the lab, right? Not necessarily a good outcome. We wanted to understand why this happened. So we had a look at their microbial community. Now, don't be overwhelmed by this picture. Essentially, what you're looking at is different coloured dots, and those dots are different treatments. What we were hoping for, if we saw an effect, is all the red ones are grouped together, all the green are grouped together, and all the blue are grouped together. You can see they're a mismatch everywhere. It means the treatment did nothing. It had no effect whatsoever when we put it in a live animal. Now, I can see a lot of you having a face of confusion, right? This is not a good result. And the reason I'm pointing this out is this is extremely common. As I said, there are 28 products that we know of as scientists, and in my professional opinion, one of them works. One, right? One is commercially available beyond the research setting, and that's what I'm going to focus on today. And some of you might be shocked because it's not seaweed. It's actually 3-nitrooxypropanol, or 3-NOP, 3NOP it might be called. Bovaer is a name you may have heard before as well. This one is really special, and it's special because it targets the enzyme that makes methane. Without it, methane cannot be produced, or it's significantly reduced. And it's really cool because it's actually metabolized in the rumen of the animal. So it works, reduces methane, it's metabolized into products that are already there. So there are absolutely no residues present in milk or in meat, which means it is entirely safe for everyone to consume. And for that reason, it's authorized for use in Australia and 56 other countries around the world. It's still in the development phase. Now it's commercially available, you can't go buy it off a shelf, but it is becoming more and more acceptable and more used commonly throughout the EU and the US and also here in Australia. Now, it's important to note we only include really, really small amounts of this. So the number on the screen is 1/4 teaspoon. It's 100 milligrams per kilogram of food that animal eats. It's like a drop in a bucket. It's nothing at all. And it works in as little as 30 minutes. The downside to this product is because it's metabolized in the rumen, it's gone quickly. So unfortunately, it lasts for 12 hours. And then you need to provide another dose or another feed to that animal, able to keep that methane reduction going. Right? Now remember that as we go through, because there are different mechanisms and different ways in which potentially that could happen, and some of that I'm going to talk about tonight. So we know this product reduces methane quite effectively. 30%, pretty much at a minimum, somewhere in 30 to 60% range is really, really good. And it does this, as I said, by blocking that production of methane by those methanogens. Now, the theory is, or has been, I should say, that if you reduce methane, that hydrogen gets redirected back into the animal and the animal can grow better. That was the theory. 50 years of research, we still haven't been able to prove this, unfortunately. Right? So we thought that's what happened. The theory I have currently is what happens if we can balance how much methane we reduce with how much hydrogen is produced. So I think if you overwhelm the system and we just reduce all the methane, but there's all this hydrogen, where does it go? It has to be burped back out, right? It can't go anywhere else. The system can't use it efficiently. But what if we limit how much methane we reduce so that there's not as much hydrogen? Maybe the system can actually account for that and redirect it back into the animal. That's the ideal scenario, right? That's what we want to see. Now, our lab is currently testing this theory, and we're doing it by administering 1/2 dose, so 50 milligrams of this 3NOP in feedlot cattle. So we're doing this in 2 different settings. We've got a research farm out at UQ Gatton, which looks like this, and we've got 48 head of cattle. Now, these are preliminary results, so we're only halfway through, don't get too excited, but I am going to share them as sort of groundbreaking results today. What's happening is the animals are coming in and they have access to these green feeders at the top. Now, each one of those has a treatment allocation to it. The animals have an electronic ear tag. When they come up, a gate either lowers or it doesn't. So it only allows those animals that have registered for that treatment to access the feed bunk, which means despite being group housed and working in a team, these animals, they're able to get an individual treatment. So we can have a lot more animals housed in that pen, but still get individual data. And we're also getting methane data out of these guys. So intake and methane. This is our controlled study where we know every single parameter has been quantified. And this is what's happening so far. Intake, as expected, wasn't affected, right? We're getting an average 12 kilograms per day. But we're getting that wonderful 28% reduction in methane. So it's working. We've halved the dose, we've halved how much we need to feed these animals, and we're still getting a 30% reduction, right? This is quite promising data. Again, fresh off the press, the trial ended Friday. So pretty cool so far. We then took this to a commercial feedlot. So this data is for 500 head of cattle. We're working up to a total of 3,000, so it won't end until January next year. Think of it as a race. We want to get as fast as we can to the end point. And the end point in this case is meat production, right? We want our animals to produce as much as we possibly can in the shortest amount of time or with the smallest amount of feed. So in this case, we're getting a little bit of an increase in weight gain, so 3 kilos, and they're eating a tiny bit less. The numbers don't seem like much, but when we convert that to an efficiency metric, the smaller number, the better. It means they eat less and they're producing more. To put that into a bigger context, our target weight gain for these animals over around 125 days is 240 kilos. Our control animals eat 28 kilos more to get to that point. 28 kilos doesn't seem like much, but per pen in a feedlot, that's over 3.5 tonne. To put some economics behind that, at an average of around 50 cents a kilo, it's $1,800 a pen. That's significantly higher for the control group when you talk about money going in and out of a feedlot. So potentially, by giving a small dose of this product, we can actually save the feedlot money by making them a more efficient system. Pretty cool results, I think. So we talked about feedlots. Feedlots are good, but they're also really easy to measure stuff. We know exactly how much they eat. We know what's in there, we saw them eat it, we can watch them do it. Grazing cattle are an entirely different story. Have you ever tried to measure intake? We did. We failed miserably. We actually found out that cattle eat more than, the grass grows more than the cows eat, right? So you measure at the beginning, you measure at the end, and the grass ends up higher. They didn't eat for 2 weeks. Unlikely, right? It's really, really hard to measure intake. In fact, impossible to measure intake accurately. And it is that, along with the fact of how on earth do you supplement them when they're out grazing? How do you make an animal come and eat something? That has led to no studies to date being published in an entirely grazing system, right? We just haven't done it. Now, me being me, and my lab being my lab, we saw that as a challenge. So the next study we're going to talk about tried to address that, right? So our first consideration was, how on earth do we do this? How do we get our animals to come in and eat this product? And we want them to eat it multiple times per day. How do we get them to have good methane data? How do we do all of these things in a grazing environment and have them give us good data? What you're seeing behind me is an image or video of a green feed machine. This is an automated piece of technology that delivers pellets as a bait, which entice the animals to come in entirely voluntary. As the animals come in, their breath that they emit is measured for methane. We don't have to touch them. All we need them to do is eat. And this was our delivery mechanism. We put our Bovaer, our 3-NOP, into our pellets, and we put it into an automated system, and we got them to come in, right? Now, that all sounds really, really easy. It wasn't quite that easy. The choice of feed really matters, right? We tried a loosened pellet, so something that is mimicking pasture. We wanted to be as close as we possibly could to those grazing animals. And don't be overwhelmed by this again. The blue bars basically measure intake. Not one of those blue bars is the same. Not exactly what we're aiming for. We want the animals to consistently come in and eat the same amount every single day. They came in for brekkie at about 6 a.m. and then they kind of snacked. There was no pattern to any of their consumption, and we want to be consistent. We want to know when they're coming in, right? So what we did was we looked at a grain-based pellet. And you can see straight away those blue bars at the top, fairly consistent, right? We were starting to get much more consistent intake, and visitation started to form a pattern. They came in almost every single day for breakfast at that 7 a.m. mark. And then really cool, every 2 to 3 hours later, they came back. So we were starting to understand the behavior of these animals, that we needed to have something really tasty, think of it like a lolly treat, to get them to come back and come back time and time again. These animals were on 20 hectares of pasture. We needed something to get them to come back, right? So in this case, our grain pellet worked really well. We kind of understood the behavior, or we thought we did. So we then moved to what we know as a backgrounding study. For those of you not familiar, the term backgrounding essentially refers to cattle that are growing before they enter a feedlot. Now, in the Australian production system, that's primarily done in grazing environments. In other areas of the world, that can also be in smaller base pens with grain. But for the context of today, entirely grazing setup. So the image I'm showing you here is what our system looked like. This is out at UQ Gatton as well. We had a setup of 6 different pens and 3 treatments. 150 head of cattle split into 50, 50, 50. We had a control where we used our loosened pellet because we have to have some way of getting a methane measurement. We just had to. We had our grain-based pellet, and then we had the grain including that 3-NOP. And we rotated them through all of these paddocks so that they all had the same consumption of pasture. We used different green feed machines to make sure there's no biasing of anything. And what did we find? Actually, some really cool data. By supplementing our animals with 3-NOP, so that red box, we saw a 19% increase in the growth of these animals compared to a straight loosened pellet or a grazing environment. And if we compare that directly to our energy only or our grain only supplementation, we were getting a 7% increase in body weight. And that's over a 56-day period. So these animals were growing considerably well over such a short period of time. And really interestingly, obviously, we want to know what happened to methane, right? They grew, really cool, but what happened to methane? We saw that standard 25 to 30% response that happens when we introduce 3-NOP. We compare our energy to our 3-NOP group, and we saw that 25% reduction. What we're saying here is for the same body weight animal, they produced 25% less methane, and they're growing 7% more. So these results provided quite an interesting aspect of how we might actually introduce this into industry, right? So to put it into perspective, we want animals to get to a certain weight as quickly as possible. In this backgrounding scenario, we want them to grow 200 kilos. Our control took 267 days to get there, our energy 241, and our 3-NOP 213. It's 54 days faster, right? That's quite considerable. That's a quarter of the time. In terms of methane emissions and carbon dioxide equivalents, we also had a reduction purely because these animals would theoretically be around for less time, right? They go into the feedlot sooner, they're around for less time. That equates to a $37 net profit. When we standardise live weight, so if we say every kilogram of body weight is $4 and it costs you $500 to supplement 1 tonne. Across all different treatments, what you end up with is $37 per animal more when you feed this product. That's accounting for the cost of the supplement as well, right? So quite a nice incentive for industry to actually be able to implement this. Now, if and when the government actually provides carbon credit scheme for this, there is an additional $12 that can be made for every head based on the carbon dioxide equivalent that you can save. So that's almost $50 a head we're coming out in front by actually adding something that costs money to this diet. Now, that all sounds well and good, but it's also really important to remember this is a research setting, right? So we had a very big luxury of using that green feed machine. It's an automated device that is quite expensive, and we have the technology to know how to use it and know how to fix it. In industry, there isn't necessarily the access to a machine that can feed these products out every 3 hours. And if you remember, in industry, those 20-hectare paddocks are now 200+ hectare paddocks, right? They are enormous. So how do we actually tackle this challenge? There's some ongoing research about how and why and different products are being tested. Water delivery is one. Lick blocks, so a block that the animal comes and consumes at will. Slow release mechanisms are another. Something my lab is really, really interested in is what we call early life imprinting. Now, to break that down for you, a ruminant animal is born with a sterile gastrointestinal system. It means there's nothing there. Everything that animal comes in contact with from the second it's born decides what resides in that rumen. Once it's a mature rumen, so once the animal is weaned off its mother's milk and it's consuming only grass, it is extremely, extremely resilient and very, very difficult to change. So the theory that we have is if you can implement something at birth, you can actually modify what happens for the lifetime of that animal. So the next study I'm going to talk about did just this. We looked, in this case, in dairy calves, and we had a control group where we put just water, and we did it orally, so a little syringe in their mouth every single day. And then we included 3-NOP in water for our treatment groups. Every day, from the day they were born until they were 14 weeks old. So let's say approximately 3 months. And we wanted to see what happened. Could we alter methane? And can we alter growth? What is happening if we treat from birth? Now, with these animals, we didn't see any effect on their growth rates. So that grey box is when they were supplemented, right? So they were only supplemented, remember, for the first 14 weeks of life. This is up to 1 year of age. So we're still seeing no effect of body weight, which is okay, right? No effect is still good. We're not negatively impacting these animals. But what became really cool was when we looked at the methane. Now, we started at 3 months, so again, that grey box is that treatment period, and we had a 10% reduction while they were treated. So a smaller than normal reduction, but remember, these are dairy calves and we're feeding it down their throat every single day. We also saw that separation of the microbes, so that's the little graph down in the bottom. So what we're seeing there is this treatment is actually having an effect on the development of that microbial composition. We're getting a separation between our treatment and our control. Now, if we have a look at 6 months of age, now remember, these animals are no longer treated. We've stopped treatment 3 months ago. We still have an 11% reduction in methane. And that microbiome, those microbes, are still distinctly different. This was quite exciting news, right? We've stopped treatment and we're still seeing that effect. What became even cooler was 9 months later, at 1 year of age, we saw an even bigger effect. Almost 18% difference in methane emissions. And that microbial community is still distinctly different. Now, that might not sound that exciting, but it's the first time that we've ever been able to do this. We've given a treatment at birth that has changed the composition of the microbiome in a way that it didn't affect growth. The animals are still perfectly healthy. But we were able to imprint a reduced methane emissions for the first year of their life. That's pretty cool. So guys, I will wrap it up there, and I will leave you with some final thoughts I have. That essentially, methane emissions are an evolutionary process that we are fighting. And I've said we've spent 50 years, or other people, not myself, have spent 50 years trying to tackle this. And we are making progress. We've got one product, right? It can work, but the problem now is getting it into industry, right? As a scientist, I'm fairly confident this thing works. But getting it in a way that we can implement it in a feedlot, in grazing animals, and have a really, really strong effect is where we're at now. That is the status of this research. And our group alone has shown low doses potentially can balance that methane with hydrogen and actually maybe redirect it back into the animal and make it a more efficient system. That's going to make feedlots adopt this a lot faster. And again, if we can supplement throughout the day, we're going to potentially be able to see more of a productivity difference in grazing cattle. How we get it into grazing cattle is another question. But I'd like you to consider one final thought. What if we could just treat an animal at birth and it would have its whole life with lower methane emissions? It'd be pretty cool, right? That's what we're working on. And that is our goal for our lab. So stay tuned. We haven't got there yet, but that is the grand plan. So with that, I'd like to say thank you, especially a big thank you to my lab who are present today, who actually did all this work, and the other members listed.
Thank you so very much, Sarah. Very shortly, friends, that QR code's going to appear again on the screen, and that's going to take you to the platform by which you can ask any questions, and it'll come straight to my phone. So go ahead if you haven't already, and if you've got questions for Sarah tonight, can you go ahead and please type them up, and we'll get through as many of them as we can. And just while we're waiting for the questions that come through, Sarah, I have a little token of our appreciation for you, but not before you tell me, do you know where strawberry milk comes from?
No. Should I know where strawberry milk comes from?
Why they come from strawberry cows, of course. Sarah, this is actually one of my designs, and I thought I'd throw in this little token of our appreciation for you. This is a cow stir. You see what I did there? And thank you very, very much.
Thank
Sarah,
you. I will treasure that.
And with that, we've got some questions coming through now, and once again, we'll try our best to get through as many as possible. And I think this is a really cool one. Do us as humans, do we produce methane?
Yes, we do. We actually do produce it out the other end. So similar to horses, our digestive system is set up in a different way. We don't produce nearly as much. So you can be grateful that your flatulence isn't contributing as much.
Very good. Here's another good question. With NOP3
yep.
being a supplement, have you measured also meat quality and other aspects of the animal's health?
Yep, so we specifically haven't. It has been done extensively within Australia, so the University of New England has done extensive studies on that as well. There's no effect. We have feedlot data that says it doesn't do anything. It really doesn't have any effect.
Wonderful. Here's, again, another question. I think it sort of follows on from your last dream and vision of imprinting from birth. And you also alluded to one of the challenges being scale. How do you envision the scaling up of that imprinting, stage?
Yeah. So it's a work in progress, let's say. It's an area that really requires very careful consideration, right? So in the dairy industry, it's quite easy. We have access to our calves from day one. We can access them and we move on. In the beef industry, weaning, so when we remove them from the mother's milk, is usually the first time those animals have contact, if at all, or maybe when they're branded. So we do have to understand, before we can scale up, at what age is the best time to do it. So the study that we did covered both birth and weaning. Potentially you can do it later in life. How we get it into day-old beef calves is going to be a challenge. Potential to feed it to the mother and have some sort of transfer. Again, the downside of 3NOP is there's no residue in the milk.
Yeah.
So that actual transfer and how we do it, that is one of our biggest challenges.
Yeah.
Yep.
So am I correct in thinking that, you mentioned that calves are born with a completely sterile gut, and they develop that over time, and typically in that sensitive period when they're young. Can we assume that has a lot of similarity to the mother's gut?
Yes. Yeah, so essentially those first 2 weeks of life are really the area that we have been targeting. Potentially later in life as well, but some other work has suggested that's not going to be as effective. So as the animal gets older and older, it gets much more closer to the mother, or like a core microbiome that almost every ruminant species has.
Cool. Here's a question from another Sarah. How is 3NOP manufactured?
Oh, I have no idea. You have to talk to DSM. They've probably got a patent out there somewhere.
Even if you knew, you probably maybe can't say.
It's a synthetic compound, that's all I know.
Yeah. Would it be possible to have the cows ingest NOP3 through the air or as a gas?
It is volatile, so if you leave it out in the sun for too long, it does volatilize. Breathing it in doesn't necessarily get it to the rumen, right? So the trick is it has to go to the rumen. So when they breathe in, it's going into the lungs. We need to ingest it, because remember, the mode of action is targeting that enzyme, and that's only in the rumen.
Yeah, right.
So even an injection, it's got to get to the rumen itself.
Right,
yeah.
Gotcha. Yeah.
That would be cool, though, if we could get it in the air.
Great.
Take notes.
Okay, I'm just going to refresh this a little bit. I think you did mention some next steps, so we'll Oh, just to be controversial, are there any foods that increase a cow's methane production?
Yeah, so as I said, the main reason cows produce a lot more methane is fibre, right? So the grasses, everything they eat on a grazing system in a pasture, they emit more than they would in a feedlot, right? So that's why I said the majority of our work focuses on that grazing environment. How do we get it into them? Because that's where we can make the most effect.
Yeah.
Now, even in a feedlot system, if you feed different grains, different compositions, are they partially broken? What is the form of those things? That can also influence methane as well.
Yeah.
So very much so, it's all diet-based.
Yeah, awesome. Okay, let me just refresh this a little again, and let's see if we can maybe do a couple more questions. How much is that feeding machine?
Ooh. I mean, I haven't bought one in a while, but it hundreds of thousands of dollars, put it that way. They're from the US, so you also got to do the conversion. You don't want to know. It's very expensive.
Yeah, awesome. Okay, we might wrap it up with a couple more. And I think you sort of mentioned that you did some of the rumen analysis and gut content analysis. How do you actually test for what's in a cow's rumen?
Yeah, so there's several different ways. I'll just put this one down.
Yeah, sure.
There's several ways we can do it. So I didn't show you an image tonight, but we do have what we call fistulated cattle. So the cattle that have a hole so we can access the rumen. It's a very common technique that can be used to gain access continually to rumen content samples rather than having to put a tube down the animal's throat is the other method we do that. So the metabolism study, we do actually collect samples every couple of hours, which means we can track what is happening with that fermentation and the digestion. We can say 2, 3, 4 hours after they've eaten the food, what is in that rumen. We take a sample of the rumen fluid, and then it does go through a lab analysis. So gas chromatography will give us the breakdown of those molecules and then what level they're present at.
Yeah. Well, thanks again, Sarah. And once again, if there are any further questions, please feel free to stay behind and have a chat with Sarah. But we will leave you with, just because we have so many younger members of the audience, Sarah, can I get you to reflect on your journey to this point in your life and maybe tell us a little bit about what it means to study agriculture in this day and age?
Yeah, I mean, that's not a short question, but yeah. So I guess my life journey to here has taken me all around the world. So I would say for those of you interested in agriculture, it is not all tractors. I haven't driven a tractor since high school when I ran over a garbage bin. That was enough for me. What we did is looking at animals, right? So I was very interested in nutrition, which is one of the things we teach as part of the agricultural degree. And that got me really excited in doing research. I then spent 4.5 years in Canada, so I still have a slight accent, sorry. And then I very luckily got to spend another 2 years in France before coming back to Australia to where I am today. And I actually teach part of the agricultural science program. So bringing in that worldwide perspective of what we learn about and our research really heavily flows into what we teach as part of that program. And from the animal science major within it, it is very industry driven, a focus on how do we improve productivity and efficiency, and then also profit, right? So we're not all about just throw stuff in and it costs you money. It's got to make you money. Because at the end of the day, that's how we actually get people to use products like 3-NOP. I can do all the research in the world, but if it's too expensive, it doesn't mean anything, right? So we do try and incorporate that in a lot of the teaching and really bring our research through in all of those lectures that I give every day.
Awesome. Thank you very much. And can we please give Sarah another round?
Thank you.
Thank you very much. Thank you.
I'll take that one. Thanks so much.
Awesome. Just a few announcements while we wrap up today. Our next Bris Science in October will feature Professor Helen Bostock. She's an oceanographer, and she's going to explore how Antarctica's changing climate is shaping Queensland and the world. Now, just a couple more announcements. The first being that sometime tomorrow, you're going to receive a follow-up email from us that includes a link to a little survey form that we would highly encourage you to complete and give us your feedback about the Bris Science lecture today. And finally, if you would like to join Sarah and each other. We have some refreshments outside for you to enjoy. And please stick around and have a chat with our speaker tonight as well. So thank you again, Sarah, and thank you all very much for joining us today. Until the next one.